Good morning, my dear friends. If you look at your regular studies, from morning you get up, go to school, study something and come back. This was your mode of studying till last year. This year, due to this pandemic, COVID-19, we have just changed our mode of studying, that we are just studying, looking at your mobiles and laptops or something like that. This actually made the teaching and learning little difficult. But what is the message we are sending to the world is that we will be studying and learning whatever happens. We will be keep studying. That is the simple message we send to this world. So before we start this lesson, let's start this one with a small story. Of course, we all like stories, am I correct? And the story is like this. There is a fighter, he always wins. Doesn't matter who is the opponent. Maybe a stronger one or a weaker, taller, shorter, whatever it is. This fighter always wins. One day, one opponent came with a helmet wearing on his head. People laughed at him. You are going to make a fight that you are definitely going to fail, and still you are wearing a helmet. What is the secret? And that fellow replied, I just analyzed my opponent, that he always start the fight with a slap on the face next to the ear. That makes the opponent lose the balance before they start the fight itself. That is his success story, that he always wins. So now I know his techniques, I know how to handle him. So even if he beats me next to my ear with this helmet, and for the first time the stronger fighter failed. That is the end of the story. I wanted to tell you one more live example that you normally do, so that you understand it well. Let's say you're going to play a cricket and with another team, and you found people are there, and you will start telling, okay, this man will go for fast bowling and this fellow's technique is that, and likewise you will analyze everybody and you will make strategies. What is their strength? What is their bowling style? How we can defend? How we can win? You will analyze your opponent well so that you can success. That is the technique we should always handle it to deal with an enemy. When we look at this COVID-19 pandemic, you may come across that stronger nations, weaker nations, strong men, weak men, kids, youth, aged, everybody is affected. So what it says that coronavirus is our enemy, we should know how to handle our enemy. Once we know how to handle these fellows, we are getting stronger. We will win. For that, you should know some basic ideas about this type of microorganisms. The knowledge about this coronavirus that we have made us to use the soap and hand sanitizers. What is the secret behind using hand sanitizers and soap to destroy this deadly, dangerous, silly thing? Use the soap, coronavirus dead. Of course, on the surface of the skin. What is the secret? Since we have the knowledge of this enemy, we are able to make this. Otherwise, we will still find no way the entire human population could have been affected. So knowing the enemy is what we are going to do in this lesson. My name is George, and today's class we will look into Unit 22, the world of microbes. In this lesson, we are going to learn things like what are the microorganisms. What are their types? How do they function? Categorize the types of virus, and what are the uses of these microorganisms. And the thing is, not all the microorganisms are dangerous. Some of them are beneficial for us. We will learn that also. How do they spread disease? How can we prevent them? All these are the main content of this lesson. When we look at the introductory part of this lesson, the first term you will have to study is microbiology. The microbiology is the branch of biology that deals with bacteria, fungus, virus, algae, and all this type of microorganisms. And microorganisms are like, they are microscopic. You cannot see them with your naked eye. You need a microscope to see that. They all are microscopic. They live in different conditions, like they can live in water. Some of them are able to live in extreme conditions of ice and hot climates in the deserts, like that. They can live inside the host of like a parasite. Like some microorganisms are living inside us. And if you look at the microorganisms, all of them are not dangerous. Some of them are beneficial for us also. Example, rhizobium bacteria, that they fix atmospheric nitrogen. And the bacteria, some of them are converting the milk into curd, and some of them are helping in preparation of medicines. All of them we will see in this lesson. And looking at their shape, they are mostly unicellular. That means only one cell. Some of them are prokaryote and few are eukaryote. The microbes may be unicellular, like a bacteria. Sometimes they may be multicellular, like a fungus. So sometimes this can also be acellular. That means there is no cell. Example, virus. Viruses, they don't have a proper cell structure that has all the cell organelles inside, like nucleus and other things are not present inside the virus. So this is a major introductory part. Some of them are beneficial, some of them are dangerous, and some of them are unicellular, some of them are multicellular, and that they can be living in almost all the conditions. So we shall start the lesson. So if you look at the world of microbes, we are going to discuss about all these microscopic organisms. If you look at that, all of them are microscopic. We cannot see them with our naked eyes. Then they can live in almost all the type of atmospheric conditions, like extreme hot climate, or extreme chill climate, or heavy pressure, like the microorganisms are living deep under the sea. All these type of conditions, or they can be aquatic, or they can be in freshwater and saline again, or in desert, or in ice glaciers. So many places they can live. So they have the specific adaptations like that. And when the moment we talk about microbes, we can't say that all of them are dangerous. Some of them are very good. For example, the bacteria that convert milk into curd is beneficial for us. Or a bacteria, they are helping us to build a vaccine or a medicine. They all are helpful for us. They are good. But some are causing severe damage to human tissues. They can be called as dangerous. They are bad microorganisms. And when you look at their structure, they can be unicellular. That means a single cell, like a virus or a bacteria. Or sometimes they can be multicellular. That means so many cells are joined together to form a single organisms, like for example, fungus. If you look at the next one, acellular, this term says acellular is not a cell at all. That means virus. Viruses are not showing any character of a living cell. We will see that again in detail. And what are the microorganisms? We are going to look at this chapter is 4 in number: bacteria, virus and fungus, and viral proteins also we are considering as microscopic plus dangerous. That's why we have included prions. So we will discuss this one by one. Let us start with the first microorganisms, bacteria. Bacteria are prokaryote. Let me rewind a small topic from the lower classes. Prokaryote means the cell organelles, membrane bound cell organelles like mitochondria, Golgi complex, lysosomes, nucleus, all these things are membrane bound organelles which is present inside the cell. If the membrane bound organelles are absent, a prokaryotic organism means cell that has so many cell organelles inside like nucleus, mitochondria, Golgi complex, lysosomes, nucleus, all these are membrane bound in organelles which is present inside the cell. But if you look at organisms like bacteria, they are called prokaryotes. They don't have any membrane bound cell organelles inside. So the most of the functions of mitochondria and other things are performed by the plasma membrane itself. Their genetic material is open, that may be floating inside this cytoplasm itself. So such type of organisms are called that they don't have a cell membrane bound cell organelles. They are called prokaryotic organisms. And the first point itself is bacteria are prokaryotic organisms. They are microscopic from 1 to 10 μm in length. That's a very small organisms. And they are just, some of them can move in a water content. If they are present inside the water, they can definitely move. They have a tail-like structure attached to their body is called flagella. These kind of small minute projections from their body, they can make them swim. So such type of bacteria are motile bacteria, and some of them are non-motile. They don't have flagella, they will float along with the water or the media content. So that is the general introduction. They are prokaryotic, some of them are motile, some of them are non-motile. They are very microscopic. That's the best starting introduction for the bacteria. And if you look at the bacteria, are they all same? No, they are not same. All of them are not same structure and shape. They have different structure and shape. Based on the shape, bacteria is classified into 3. There some of them are spherical shaped called coccus. Second type is rod shaped. We call them as bacilli. And the third one is spiral kind of things. They are called spirilla. These are the 3 different shapes of bacteria is concerned. If you look at the history of microbiology, in the year 1674, Anton van Leeuwenhoek, he just observed a part of his teeth he observed under microscope, and he found that minute organisms are moving. And that is the first initial stage of started observing microscopic organisms. In fact, that was the first time people started to show some interest in learning things which are not visible to our eyes. That was the beginning in the year 1674. If you look at the structure of a bacterial cell, bacterial cells are mostly covered with a rigid, strong cell wall. In some species of bacteria, they may have one more protective layer around their normal cell wall. They are made up of polysaccharides. They are called capsules. Sometimes the bacteria may be living in an extreme cold temperature or under deep side of the sea where the pressure is very huge or temperature is very huge or high or low. In these kind of extreme climate conditions, these bacteria may have an extra covering on their bodies called capsules. Most of them are made up of polysaccharides. And when we look inside the bacteria, these bacteria are prokaryotes. We discussed that earlier. Since it is a prokaryote, they don't have any type of cell organelles inside. The genetic material is DNA and few ribosomes inside this cytoplasm, and that is a major part of this cytoplasm itself. So inside the plasma membrane, cytoplasm, that has few ribosomes and a genetic material. It's a DNA. That is the only content you can see inside the bacteria. So inside the plasma membrane, you have a DNA as a genetic material and ribosomes. Both of them are working together to build proteins, protein synthesis. In few of the bacteria, you will see an extra chromosomal. That means the main DNA is there. Apart from that, a small circular shaped genetic material is still present inside the cytoplasm. That kind of small genetic material, apart from the main one, the circular small shaped genetic material is present. We call it as a plasmid. So inside the bacterial cell cytoplasm, that has a main genetic material, ribosomes, and sometimes you may see a plasmid, a extra chromosomal circular DNA. That is the introductory part of the structure of bacteria. Let's check this one more time so that you will find a little clear understanding of the structure of a bacterial cell. Let's start with the first microbe of this lesson, bacteria. They all are microscopic. That is the first point. You cannot see them with your naked eyes. Next one is they are prokaryotes. Prokaryote means they have a single cell, that they don't have any of the membrane bound organelles inside. Their DNA it is floating inside, and most of the functions of a mitochondria and other things are performed by the plasma membrane itself. Such type of cells that they don't have any kind of membrane bound organelles inside are called prokaryotic. So bacteria is a prokaryotic organism. And coming to the size of the bacteria, they are 1 to 10 μm in length. Some of them are motile, that means they can move. Some of them are non-motile, they cannot move. So those times they may use the floating of the water. Along with the floating of water, they will move, or the medium. So they are non-motile. Let's say they are motile, they have a tail-like structure called flagella. And when you look at the shape of the bacteria, all of them are not the same in shape. Some of them are spherical, we call them as cocci, and they are spherical in shape. Some of them are rod-shaped, they are called bacillus. So rod-shaped ones. Some of them are spiral in shape, we call them as spirulum, they are in spiral in shape. So based on the structure of shape of a bacteria, they are into three types: spherical, rod-shaped, and spiral. And when you look at the structure of bacteria, this is available in your textbook. You have to make a copy of this into your fair note also. So since it is a prokaryotic organism, they have a cell membrane. Inside they have genetic material and cytoplasm, and a small extra chromosomal circular DNA. We call them as plasmid. And this plasma membrane alone is not so strong, so they need to have an extra strong covering. We call them as cell wall. And above that, in extreme conditions like these bacteria is supposed to live deep under the ocean with a heavy pressure on it. They are supposed to have a strong capsid, a strong coat around it. So capsid inside cell wall, and inside that you got plasma membrane, and inside that cytoplasm and a chromosome, plasmid, and few ribosomes for the protein production. That's all the structure of a bacteria we can discuss. They are not at all complicated. This is the general introductory part of, and in higher classes you will study various types of bacteria and all. This is just an introduction part of this bacteria. The next one is the virus. When we look at the next member of this world of microbe, that is a virus. Virus, the name in the Latin language, that means venom, poison. These microbes, the virus, are just acellular self-replicating parasites. Meaning is acellular in the sense you have studied about the cell structure in the previous classes, that a cell has a cell membrane, cell organelles inside like nucleus, mitochondria, Golgi bodies, all these things are inside. But acellular means this virus does not look like a cell at all. They don't have any plasma membrane, they don't have Golgi bodies or any of this kind of cell organelles inside. Instead, these fellows have an outer covering made up of proteins and a genetic material is packed inside. And these proteins are the only structural part apart from this chromosome, maybe a DNA or an RNA. These are self-replicating. That means they are able to make their own host, their own copies when they are present inside the host. So when we talk about the virus, we say they are self-replicating acellular parasites. What is the meaning of parasite? A parasite is nothing, an organism that depends or live inside the host cell to absorb the nutrition and protection, all this from the host for its well-being, multiplication, for the food and all the other things. And they are just parasite. They always need a support of a host. So that is why a virus is called acellular self-replicating parasites. The next part is when we look at the structure of a virus, just a protein cover, and that has a genetic material inside. Maybe a DNA like T4 bacteriophage, or a genetic material can be RNA like a tobacco mosaic virus. And if you look at the size of that in nanometers, they are in 18 to 400 nanometers in length. So these viruses always need a host. That host can be a plant or an animal or a bacteria itself. This virus can live inside a bacteria because the bacteria is very bigger than a virus. Then you can imagine the size of a virus, how small it is. Transmission of one virus from one host to another host, it is easily happening always, because this virus can spread through various mechanisms, maybe through a cough, maybe by a touch, or by the body fluids like sweat or something like that. So the easy transmission of this living organism is a plus point for this viruses. Of course, it is a minus thing for all the other animals. When we look at this virus, they have a specific capacity that they shows both the character of a dead organism and a living one. So we can say that this virus actually stands between the living and dead. Why we call them so is because viruses are showing when they are outside the host, they are just powder, just a facial powder like that, dead. Once they are inside the host, somehow they get inside the host and they will start behaving like the living one, like reproduction, eating and excretion, cell replication, all these are happening inside. So virus acts as both alive as well as dead. If you look at the live characters of a virus, we can say that they have a genetic material like what we have, maybe an RNA or DNA, that depends. And they replicate when they are inside the host, they reproduce. And if you look at another aspect, that how the virus, we can call them as dead, because when the virus is outside the host, they are just inert material like a stone or a facial powder like that. It is very inert and material, so it looks like dead. Next point, how we can say that this is a dead material, is that they don't have any type of cellular organization inside, like cell organelles absent, plasma membrane absent, cytoplasm absent. They have just a genetic material packed inside, that's all. And one more point is that these viruses, when they are outside the host, it can be crystallized. You know, the crystals are the purest form. You may have studied in chemistry. So these things can be crystallized. So let me conclude that these viruses are showing the characters of both living as well as dead. Okay, now what are all the viruses? Okay, now if you have a question, all the viruses are same? No, not all the viruses are same. In fact, viruses are classified into three based on the mode of in which host they actually attach. The first one is plant virus, tobacco mosaic virus, cauliflower mosaic virus. All these things will infect only the plant. They don't infect the human beings, or they don't infect any other animal like that. And next one is animal virus, like polio virus, HIV virus, and all these things virus which can infect animals like human beings and all, they can infect. That's a specific type of virus. These viruses will not infect the plants. And the third type of virus is bacteriophages. The name itself says actually what it is. I told you the bacteria is far bigger than a virus. So definitely a virus like T4 bacteriophage, like viruses, can infect the bacteria. So a bacteria also kind of getting a flu or a fever like that. So bacteria also getting infected with bacteriophage, another type of virus. When we look at the types of virus, they are into three: animal virus, plant virus, and bacteriophages. So when we look at the structure of a virus, that is only has a protein cover and a genetic material inside. But we are going to talk about one more thing, that without this genetic material inside, this protein part outside alone can make infections. They are called virions. And the pathogenic, dangerous genetic material, RNA inside, that's genetic material itself, can penetrate and to the plants and cause disease. They are called viroids. So these are the 2 points you should remember. And if you look at the structure of the virus, let's talk about the next member of this lesson, that is the virus. If you look at the details of virus, in the Latin name, the meaning itself is venom, the poison. And these viruses are non-cellular. That's the first point you should remember, is because you remember the structure of a cell that has a plasma membrane, cell organelles are inside, but this fellow don't have any of these things. That is the meaning of non-cellular. But then what do they have is just a protein cover and a genetic material inside. That's all. That is why it is called as non-cellular. They don't behave like a cell at all. They are just self-replicating parasites. What is the meaning of that? Self-replicating is once these viruses are inside the host, they will just take the replicate copy of itself. They will multiply. Once they are outside, they cannot do like that. That is why we are called self-replicating parasites. Parasites are nothing but the living inside another living host. They are all parasites. Coming to the next point, they are just 18 to 400 nanometers in length. Very, very, very small even than a bacteria. And they are shows both living as well as non-living characters. This is an interesting part. This virus can be said like they act between the life and death. Reason is sometimes these fellows behave like they are alive, sometimes they behave like they are dead. How is it possible? Because once these fellows are inside the host, they look like they are living, like they shows the reproduction, they take the nutrition inside, they kind of eating, and they transmit from one host to another host successfully. These are the characters of living things they are showing it. But sometime when these fellows are outside the host body, they are just inert materials like a stone or a piece of wood like that. They are just inert materials, just powder like they are. These viruses can be crystallized. You know what is the meaning of crystal? The purest form of anything is crystal. You have studied that in chemistry. So they can be crystallized and they just like an inert material. Sometimes if you look at their structure, they don't even have a proper cellular setup. I told you a cell has a nucleus, ribosomes, lysosomes, Golgi bodies, mitochondria, so many things are inside, but these fellows don't have anything. So in such a way, this virus are showing the characters of both living and non-living. And if you look at the virus, are they all same? No, they are not same. Based on the type of host it is attached with, the parasitic mode of nutrition, we can say them as some of them are animal virus. They attack only the animals. Some of them are plant virus. They attach to only the plant. Example, tobacco mosaic virus. Some of them are bacteriophages that they infect the other bacteria. T4 bacteriophage. If you look at the animal viruses, they are just human immunodeficiency virus, HIV, poliovirus, rabies, all these are animal viruses. And if you look at the structure of this virus, just a simple one, they have a protein cover and a genetic material inside. That's all, nothing else. So this genetic material can be of two types. Sometimes it may be an DNA, like in T4 bacteriophage. Sometimes the genetic material is RNA, like tobacco mosaic virus. So it varies. So what we have to learn from this is, when you use a soap or a alcohol based sanitizers, this protein cover, glycoproteins, can have holes in the middle. So this makes the virus to die, because this soap and alcohol will dissolve this cover that is exposing the genetic material, it will be easily dissolvable. That is the secret behind we are using all these hand sanitizers and the soap solutions to kill these viruses. And we shall see the next topic, member of this world of microbe, is the fungus. We shall see that next. The next microorganism we are going to discuss in this lesson is the fungus. Fungus is a common name. We all hear a lot of things. Fungus is just a common name. You may have seen this in many places. For example, if you let a piece of bread outside for a few days, you can see there are some patches on it, some color changes on it. If you look at that color changes through a magnifying glass, you can see that there are some kind of growth. They are called fungus. If you leave some food items like vegetables outside, when the moment it starts decaying, if you look at that, you can see some patches. Those are called fungus. These fungus are always parasitic or saprophytic. Reason is they don't have any chlorophyll. You know what will happen if there is no chlorophyll? No chlorophyll, no photosynthesis, no food production. So it has to depend on somebody else. So these fungus are of mainly following two type of food mechanisms. Like some of them are parasite, that they always look for a living host so that they can attach to it and live. And some other are saprophytic, in such a way that they always depend on dead and decaying matter to absorb their nutrition out of it. And if you look at the body of a fungus, it's called thallus, a single line. These small thallus will join together to form a mycelium, a joint structure. These type of myceliums will again join to form a hyphae. It's nothing but a tube-like structure. So inside the tube, they have the cytoplasm and nucleus present. So you have food granules, oil droplets, and other cell organelles. What are the other general cell organelles? Like mitochondria, Golgi bodies, lysosome, ribosomes, all these things are present inside that tube called hyphae. To get a strong protective outer covering, these hyphae is made up of chitin or cellulose, kind of strong fibers that is giving the strength for the hyphae to stand straight and hold the entire protoplasm inside. These hyphae are 5 to 10 μm in their length, and these fungus are non-motile. That means they can't move. They don't have any flagella, what we have discussed in the case of bacteria. So these fungus are non-motile. They cannot move from one location to another by its own kind of movement. And when we look at the reproduction in fungus, it is like these fungus are following mainly three methods. That vegetative propagation, like the vegetative part of their body itself can be cut into two and it can form two daughter fungus, like that. So they are of, the methods involved in vegetative reproduction is they can go for budding. Budding, one small organism and above that one small bud, and the bud is enlarging in size and they will detach and both of them will go for an individual organism. Or they will go for fragmentation. The hyphae, the entire setup, will go for fragments into pieces. Each individual piece will develop into a new fungus. That is another mode. Or in some other cases, this fungus will reproduce just by binary fission. Just one fungus divide into two and each two of them will become an individual fungus. So this fungus can reproduce vegetatively by three methods. That is by binary fission or by budding or by fragmentation. And if you look at the next mode of reproduction in fungus is asexual method. Asexual method means there is no male or female involved in it. These fungus will make spores called conidia. Let me tell you one general example of what is called a spore. A spore is a piece of an organism, a successful generative cell packed inside a strong shell, because this spore will look like a powder, a pollen grain. It will be carried away to a long distance and it will fall somewhere. If the situation is favorable, that time this strong shell will be broken and this new fungus will grow from that. Let's say the situation is not favorable. Because of this strong shell, without getting any type of damage, this cell will remain same there for a long time. It will wait for a long time so that a favorable condition come, so that it can start growing. So that the spore formation is the asexual mode of reproduction in the fungus. When you look at the third mode of reproduction in fungus, that is called reproduction through sexual method. In this case, both male and female gametes are involved. So male and female gametangiums are involved. Gametes are involved for the reproduction through sexual method. The male gametes are called antheridium and female gametes are called oogonium. Both they join together to form a single cell which will develop into a fully grown fungus. So these are the three methods of reproduction in fungus. They reproduce vegetatively, they reproduce asexually, and they reproduce through sexual manners. The next member of the world of microbe is fungus. If you look at the fungus, they have no chlorophyll in it. That means they have to depend on someone else for their food. Some of them are showing parasitic mode, that they will attach to a living host. Some of them are saprophytic in nature. That means they will attach to some dead and decaying body materials, and thus they will get the food from the dead and decaying. If you look at the body of the fungus, they have something called a thallus. These thallus will join together to form a mycelium. These type of many myceliums will join together to form a tube-like structure called hyphae, which are 5 to 10 μm in length. This will show a tube-like structure that contains all the protoplasm inside. This tube is 5 to 10 μm in size, and they have all the materials like all their protoplasm inside. That is nucleus plus all the cytoplasm, all the cell organelles. They have food materials also stored inside, and this one is a tube-like structure. To maintain its structure and shape, they are made up of thick cell wall made up of cellulose or chitin. This is the initial state. So these will keep growing and you will get the full-sized fungal structure. And when looking at the reproduction of this fungus, when you look at the reproduction of this fungus, they follows three type of mode of reproduction. First one is vegetative mode. That is, vegetative part of the body will divide to form new fungus. First one is binary fission. Just divide 1 into 2 and you get 2 fungus. That's all binary fission. Just divide into 2, you get 2 daughter fungus. Or go for budding. Just a fungus, go for bud, and this bud will keep growing and it will detach. That is a mode of reproduction, going for budding. Or the next one is fragmentation. At this third fragmentation technique, the fungal structure will divide into fragments. Each fragment will grow as a new fungus. That is a vegetative mode of reproduction in fungus. If you look at the reproduction by asexual mode, the next one is they will form the spores. What is the meaning of a spore is, due to unfavorable conditions, these fungal cells will make like a capsule and a cell will be inside. This is such a strong coat, it can withstand in extreme conditions, and this will be flying just a small one, just like a powder. It will be keep flowing. It can be going through water, air, or it will go some other place. When the favorable condition comes, this will just start growing as a new fungus. So this is a mode of reproduction by formation of spores. When you come to the sexual mode of reproduction, male and female gametes, Antheridium and Oogonium, they will join together to form a new fungus. They will start growing. So this is also a mode of replication, mode of reproduction in the fungal growth. So let me rewind this. Reproduction in fungus is in three styles: vegetative, asexual, and sexual mode of reproduction. And we shall see the next one. In this one is the prions. So far we have discussed in this chapter about microbes. They are bacteria, virus, and fungus. And one more thing we are going to discuss about the prions. What are prions? First, let me recollect, to answer this, let me recollect a few parts from this. The structure of a virus. Virus is made up of a protein shell and inside it is packed with a genetic material. Let's say you have this protein molecule alone, protein covering alone. Prions are viral proteins. So we have seen the structure of a virus that this virus has a protein cover. These protein and other proteins produced by the virus itself, if the genetic material is not there also, it is quite dangerous. These prions are viral protein particles. So you know the structure of virus, we discussed already that a virus has a protein cover and inside this things are packed. So without this genetic material also, this protein cover or the protein content of the virus are dangerous. These proteins can interact with the human nervous system. I'm talking about the human alone right now. This can alter our nervous system. So it is a kind of poison. These proteins that destroys our proteins are very dangerous. They normally interact with the nervous system of human beings. So prions are also dangerous microscopic things that they affect human beings. That is why we have included this as a fourth member of the microscopic organisms. Of course, this is not a complete organism but a part of the organisms. So far we have discussed four types of microscopic things: bacteria, fungus, virus, and prions. And what is their general structure? How do they reproduce? All these are the things we have generally finished discussing. The member of the microbe we are discussing is prions. In fact, they are not the entire microorganism. Instead, they are just microscopic things, the viral protein remainings. So you know that a virus that has a genetic material as well as a protein cover. This protein part alone, which is the cover or the proteins produced by the viral genome itself, these proteins, they are capable of interacting with the nervous proteins, the proteins which are present in the nervous tissues of the host. Maybe human being. If you consider the human being, these prions can interact with the host proteins, especially the nerves proteins. So instead of actual proteins which is required for the nervous system, these duplicate proteins will interact. That will actually degenerate the entire nervous system and nervous tissues, which is a dangerous thing. That is why we have included the prions, the part of viral proteins that are dangerous for the host cells. So before we conclude this lesson, what is the message you have to take today is We have learned a structure of a virus that has a protein cover which can be easily destroyable using or dissolvable by using a soap or an alcohol. So stay safe, wash your hands and stay healthy and safe. What is the next thing I would like to tell you is we have our own mechanisms inside to fight these kind of deadly dangerous things or the microbes. We call them as generally immunity, and the WBC cells inside our blood is playing a major part in it. To support this immunity, you should always take good quality food that is rich in vitamin C and D like that. And remember, the food that is in good quality may not have good taste. So whatever your parents are giving, start eating all the vegetables and quality food always. Don't always look for the taste alone, and because of the taste, don't hesitate to eat it. Stay safe and stay healthy. We will see you in the next class. Thank you.